Hey guys, my name is Shreyas and welcome to Simple Biology. In this video, we're going to be really looking into depth what ribosomes are and what they do and everything you need to know about ribosomes. So let's get right into it. So the first thing that we should talk about ribosomes is their structure. You know, what are ribosomes made out of? So ribosomes are made out of two biomolecules. They're made up of ribosomal RNA and then they're made up of proteins, okay? These are biomolecules. Just like how a table is made out of, wood, in the same sense, ribosomes are made up of ribosomal RNA and proteins. Okay. Now, in terms of the two subunits, the big parts of a ribosome, you have what is called a small subunit, and then you have something which is called a large subunit. Let me show that to you in our diagram here. This is a small subunit, and then this is the large subunit. And this kind of makes logical sense, right? Because you can physically see how the small subunit is the same size, is smaller than the large subunit. It just makes logical sense. Now, the most important thing, if you had to take one thing from this video, I would have to say this is by far the most important thing about ribosomes. What the heck do ribosomes do? Ribosomes takes messenger RNA, which is genetic information, and they use it to create proteins. So important. I'm going to even circle this because it's just so critical to know. Ribosomes make proteins. Ribosomes make proteins. Very important that you know that. Okay, and this process through which ribosomes. Okay, let me just write this out for you. So basically, how this happens is that you have the DNA. The DNA is in the nucleus of the cell. Okay. And then that DNA is going to be converted into mRNA through a process called transcription. No, not that transcription. I'll just write that down. Transcription. And that mRNA is going to be taken to this ribosome, and the ribosome is going to convert that into proteins. Okay. And the process of how this mRNA is going to be converted into proteins is called translation. Okay. So if you ever see that term, that's what it's referring to. Now, this whole process of how DNA is converted to mRNA and then is converted into proteins through ribosomes is basically transcription and translation. We'll be talking about that process way down the road in, I think, Unit 4 of our course. And it's actually something you need to know for AP Bio, this whole extensive process of transcription and translation. But for now, just know if you ever asked what translation is, this is essentially a brief overview of what translation is. Okay. Now let's talk about the different types of ribosomes. There's two types of ribosomes. There's actually free ribosomes and bound ribosomes. Let's first talk about free ribosomes. So free ribosomes are located in the cytosol. Okay. They're located in the cytosol, and they manufacture proteins that will function in the cell. Okay. Manufacture proteins that will function in the cell. So if you look at our diagram here, we have the nuclear membrane right here. We have the nuclear membrane there, which can also remember that. Oh my God, that's not the nuclear membrane. I meant to say that is the cell membrane. Sorry. So we have the cell membrane right here, which can also be called the plasma membrane, and then we have the here we have the nuclear membrane. I got confused there. We have the nuclear membrane, also known as the nuclear envelope. I'm just going to write nuclear envelope. Okay. Okay, and the region between that is the cytosol. So in the cytosol is where you're going to find these free ribosomes kind of just floating around. Okay, you saw these red dots here. Those are just free ribosomes. Now, why do free ribosomes manufacture proteins that will function in the cell? So kind of think about this. Okay, I'm just going to take a close-up of what's going on here. So let's just say we're looking at that, right? I'm just going to blow that picture up. Okay, right here. So here we have we have a ribosome, right? We have a ribosome, and this ribosome is going to just manufacture, let's just say, an orange protein. Okay. So you can kind of think of this. These orange proteins just start to form, right? All throughout the cell, these orange proteins, they just kind of form like this. and they have nowhere to go, right? They're just kind of just floating around in the cytosol. Okay, they're not going to go anywhere. As they're formed, they're just going to sit right next to the free ribosomes that they were formed. So because of that, free ribosomes manufacture proteins that will function in the cell. Now, in comparison to that, you have bound ribosomes. So bound ribosomes are located on the endoplasmic reticulum and the nuclear membrane, or the nuclear envelopes, same thing. So let me use my diagram here. We have the, if you just use a different color to do this. Excuse me. Let's use purple. Okay, so let's say, if you look here, we have the nuclear membrane here. The nuclear membrane. And then we also have the rough endoplasmic reticulum, the rough ER. Just want to point this out, even though this is kind of not relevant. This is the smooth ER here. And the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum is that the rough endoplasmic reticulum have bound ribosomes. That's kind of a mouthful, okay? So what's the difference between free ribosomes and bound ribosomes? These bound ribosomes make proteins that will manufacture outside the cell, and they also manufacture membrane proteins, which are proteins that will remain in the cell membranes, kind of just attached. Okay, so that probably does not make any sense. So I'm just going to go and show you this whole process of how a bound ribosome on the rough ER is going to create proteins which are outside the cell, as well as proteins which will remain membrane proteins, which will remain attached to the cell membrane. So let's just think of it like this. Okay, so we have the rough ER here, and then imagine that they're going to go ahead and create red proteins, the bound ribosomes. The bound ribosomes on the rough ER are going to create red proteins. So you start to see this formation of red proteins. Okay. Excellent. So what will happen is that all these red proteins are going to be, the rough ER will take it and they will, through pinocytosis, if you don't know what this term means, go watch our video on bulk transport. That's not. Go watch our video on bulk transport, because we get into, we really talk about what that is, okay? What pinocytosis is. But through pinocytosis, they're going to create a vesicle, okay? You get a vesicle, and it has these proteins, okay? And then these proteins are just kind of floating around. And then what will happen is that this vesicle is going to fuse with the Golgi apparatus, okay? The Golgi apparatus. So it'll fuse with the Golgi apparatus, and then this Golgi is going to go ahead and it's going to package and sort, and then create another vesicle through pinocytosis, and that vesicle will travel to this Golgi apparatus here, and the same thing will happen, more packaging and sorting, and then another vesicle will be formed through pinocytosis, and on and on and on like this, until we finally get a vesicle, another vesicle, right? So there's actually two vesicles that might be formed, okay? One like this and one like this, okay? So in this vesicle, you might have proteins which are just kind of floating around like this, okay? Okay, which are just kind of floating around in the vesicle. Then you also might get a vesicle which has the proteins which are attached to the membrane of the vesicle. Okay, you can see like they're actually physically attached. Actually, let me draw that even bigger for you here. It might be hard for you to visualize. Okay, so we have the vesicle here, and then we have the proteins actually physically attached to the membrane of the vesicle like this. So what will happen is that both of these vesicles are going to go ahead and they're going to fuse with the cell membrane. This right here is the cell membrane. Cell membrane. Okay. So they'll fuse. So when they fuse this vesicle, when this fuses, the proteins which are inside the vesicle are kind of just going to be just dumped out. Okay? They're going to be dumped out like this. And those are the proteins which will function outside of the cell. Now, in this case, with this vesicle over here, this vesicle is going to fuse out again, just like the first vesicle. You're going to fuse out. Except what will happen is, because since these proteins here are actually physically bonded to the membrane of the vesicle, when this the membrane of this vesicle fuses with the cell membrane, what will happen is that the proteins which are attached to this vesicle will kind of fuse with the cell membrane and still be attached to the cell membrane. So what will happen is that instead of these proteins kind of just like floating off into wherever it needs to go, like in this first scenario over here, this scenario will lead to membrane proteins which are bonded to the cell membrane. Okay, and those proteins are called membrane proteins. Okay, I hope that process makes sense. Now, the final thing I want to talk about, the last note here, is that bound ribosomes and free ribosomes are not entirely distinct. They can readily switch roles and positions. So what does that mean? Okay, so imagine you have a here again we have the rough ER here, and not specifically there. We have the rough ER right here, right here, you know. So on that we have bound ribosomes. So what can happen is that a bound ribosome here, let's just say that bound ribosome, can just decide and it can just pop off, and then it can enter the cytosol, okay, and it can become a free ribosome. The same thing can happen vice versa. You can have a free ribosome and it might just attach itself to the ER and become a bound ribosome. So they're actually bound ribosomes and free ribosomes are on a structural level, they're exactly the same thing. They have a large subunit and small subunit. And because of that, they can readily switch roles. A free ribosome can become a bound ribosome, or a bound ribosome can become a free ribosome. Okay, that's all you have to know about ribosomes for now. I hope this video helped. Ribosomes are as simple as that.